Dear students, may Almighty's peace be upon you. So, we have come to the end of the lesson, asexual and sexual reproduction in plants. At the same time, I would like to tell you one thing. Now, we hear that everywhere the death rate due to COVID is increasing. We need not to worry about COVID-19 if we have self control, or if we have immunity power, or if we are following good diet, then sure that we can overcome this COVID-19. I would like to tell you one particular thing in this video which we should follow. And my dear students, it is not that I am teaching something other than what's usual. So it is just what we say as breath exercise, pranayama. So it's a breath control we say, isn't it? So we know that it is pranayama is a very good exercise for purifying our lungs. Our lungs should be purified. So if it is so, COVID-19 will not be affected. Sure it is. So what we can do, just I'll tell you, we are inhaling through one nostril, holding it for a while, and exhaling through another nostril. This is half cycle. Then again inhaling through one nostril, the same nostril, and exhaling through the another nostril. So I'll just tell you how to do. Just you see. So I'm inhaling through this nostril. Okay. This is one cycle. So 10 times at least morning time after your prayers, you just practice it. So it will be very good for you. Okay, just inhaling through this. So close this nostril at that time. Inhale through this nostril. Hold it for 5 seconds. Then close it. Exhale through this nostril. Okay. So now, so again inhale through this nostril. Inhale through this nostril, hold it for 5 seconds, close it, exhale through this nostril. This is one cycle. Likewise, at least 5 times, maximum 10 times you do. That is a very good relaxation, very good medicine for this COVID-19. Okay, my dear students. Now let's jump into our topic. So earlier we were discussing about the pollination and then fertilization. So fertilization, fusion of male and female gametes, double fertilization we saw, triple fusion we discussed. After that what happens? In our first class itself we discussed like for the fertilization, when we say a flower, it is a fertilization is there, but before fertilization, pre-fertilization is there and post-fertilization is there. So pre-fertilization, it is development of gametes, our pollen grain and also ovule, how it is formed, everything we discussed. So during the fertilization, how the fertilization occurs, the entry of the pollen tube, all we discussed. Now post-fertilization, now fertilization is completed. So what happens after fertilization? Post-fertilization changes we can say. So when you see post-fertilization, some changes occur. In post-fertilization, some changes occur. That is your sepals, petals, your androecium, dendroecium, that is your stamens, stigma. All such things will fall off. Okay, your sepals, petals, or so during the after fertilization occurs, then all these parts, sepals, petals, stamens, stigma, all will fall off. Okay, now everything fell away. Now, what is the next thing? Then we have ovary, isn't it? Ovary becomes the fruit. Ovary becomes the fruit. The total ovary becomes the fruit. So, inside the ovary, what do you have? Yes, ovule is there, isn't it? So ovule. So this ovule becomes the seed of the fruit. becomes the seed of the fruit. Ovary becomes fruit, ovule becomes the seed. Now you think about the ovule. In ovule, what are the parts we discussed? In LS of ovule, we discussed so many parts, isn't it? So what are the parts? Funicle, the start of the ovule. Now this funicle will become the start of the seed. Okay? So it was the start of the ovule earlier. Now it is start of the seed. Okay, understand that. The next one, micropyle. Micropyle. So micropyle, what is micropyle? Where will be micropyle? The top, the opening in the ovule. Now this micropyle becomes the micropyle of this seed. Okay, so you just imagine. So the seed only it will be germinated into another plant. So for that, oxygen is needed, water is needed, all should enter into the seed, isn't it? So how will it enter? That is the question mark, isn't it? So God has created in such a way that the micropyle of the ovule becomes the micropyle of the seed, so that the water, oxygen, the entry becomes easier. So the micropyle. The next integuments. Integuments. So integuments, what are integuments? They are the covering layer of the ovule, isn't it? Integuments. So the outer integuments, integument becomes testa. Understand the word testa? Testa is nothing but the outer coat of the seed. Outer coat of the seed is testa. So the outer integuments become testa, and inner integuments, yes, you would have guessed, isn't it? So outer integument has become testa, the outer seed coat. Same way, inner integument becomes the inner seed coat, inner coat of the seed, which is called as tegmen. Tegmen. Okay, so integuments becomes the seed coats. The outer integument becomes testa, inner integument becomes tegmen. Then next one, we have synergic cells, then antipodal cells, all will degenerate. They all will degenerate. So it will not be. But at the same time, what is that? Synergid, antipodals, what is present in between? In LS of ovule, the two cells, of course, polar nucleus, secondary nucleus. Now, in our earlier class, we discussed that those polar nucleus will be fused with one male gamete, isn't it? Therefore, together it becomes 3N. So, it becomes the endosperm. So, the polar nucleus becomes the endosperm of the seed. So, these are the changes occurring, post-fertilization changes, we can say. Now, let's discuss about the endosperm. How will be endosperm? So, when we speak about endosperm, so particularly when we speak about endosperm, so there are, we know very well that how is endosperm formed, isn't it? Primary endosperm nucleus, we say, PEN. It is formed by the fusion of polar nucleus and the male gamete. So 2N plus N becomes 3N. 3N number of chromosomes will be present in endosperm. That we know already. Now, so, and we have discussed in our earlier class itself, this endosperm is responsible for the nourishment of embryo. So we discussed already, God prepares everything in such a way to give for the upcoming generation. Of course, here also for the development of embryo, this endosperm gives nourishment, nutrition to the developing embryo. So endosperm plays a major role in a seed. At the same time, this endosperm, they are classified based on how they divide. They are classified into 3 types. One is nuclear endosperm. Nuclear endosperm. This nuclear endosperm is present in arachis. You remember what is arachis? Groundnut. Groundnut. Okay. So nuclear, what happens? You imagine that this is the endosperm. So the nucleus is there. It will divide mitotically. So one cell will become two. Then again that will divide mitotically. But only thing is there will be no cell wall formation between these nuclei. That's very important. So no cell wall formation here. That is the nuclear endosperm. Do you understand? So the nucleus will divide mitotically. So many nuclei are formed. Even then there will be no cell formation. Okay, this is nuclear endosperm. So opposite to this, we have cellular endosperm. Cellular endosperm. Cellular endosperm is nothing but, so the same, it will divide, the nucleus will divide. At that time, what happens? Cell wall formation occurs. Cell wall formation. So cell wall formation. So the same thing, but cell wall formation occurs in cellular endosperm. Do you understand? So the same thing here also. We have one more type called helobial endosperm. Helobial endosperm. Helobial endosperm means, you imagine that this is the micropylar end. So now the cells will divide. So two cells will be there, isn't it? So now one will be in a larger portion at micropylar end. At micropylar end. One nucleus will be in the micropylar end. Another one will be in the chalazal end. But this is the larger portion, and this is smaller portion. So this larger portion and smaller portion. So now again this will divide mitotically. This will be divided mitotically and many cells are formed. But in chalazal region, it may divide or it may not divide. It may divide or it may not divide. That is in the chalazal portion. And even if it is so, there will be cross walls among these cells. So you can see the pictures clearly. So now, nuclear endosperm, we discussed the example as arachis. That is our groundnut. Cellular endosperm example is helianthus. Helianthus is sunflower family. So cellular helianthus. And helobial, if you see, our paddy, coconut, all such things will be in helobial endosperm. Paddy, coconut, castor, all. So now, my dear students, based upon the presence of endosperm, we have classified seeds into two. One is endospermous, that is seeds with endosperm. If endosperm is present in larger amount, okay, it is present, they are said to be endospermous. And if it is not present, they are called as non-endospermous. For this we have another name also. For endospermous, this is albuminous seeds, and this is X-albuminous seeds. Okay, albuminous seeds, X-albuminous seeds. So the seeds with endosperm are called as endospermous seeds or albuminous seeds. Seeds without endosperm are called as non-endospermous or X-albuminous seeds. So if you see the examples for endosperm, okay, maximum endospermous will be in monocots. In monocots we can see, and in dicots we can see non-endospermous. Because in dicots, now you would have got a doubt. So we said that endosperm only gives nourishment. That nourishment, it is present in endosperm. If it is not present, that is why two cotyledons are there in dicots. So in the cotyledon, the nourishment will be present. So they are non-endospermous. Do you understand? So pea, beans all come under non-endosperm. At the same time, maize, wheat, all come under endosperms. Okay, so this is the classification based on endosperm. I forgot to tell you about ruminate endosperm. So ruminate endosperm. So it is not a type of endosperm, but the thing is the surface will not be even, some unevenness or irregularity. Okay, so it will not be even. Regular pattern will not be found on the surface of the seed. Okay, so they are said to be ruminate. In endosperm itself, the surface will not be even. They come under ruminate endosperm. Now let's discuss about the functions of endosperm. Functions of the endosperm. So now tell me, what are the functions of the endosperm? Of course, as we were discussed earlier, it gives nourishment to the embryo. Nourishment to the embryo. This is first one. Then second one. So this only if endosperm develops, then only the zygote will be developed. So endosperm will be developed before. So the zygote divides only after endosperm is developed. Okay. Then third one, if you see, it regulates the precise mode of embryo development. How the embryo develops, everything it regulates the mode of embryo development. So these are the functions of endosperm. Now the next is how the embryo forms as the seed. So how it is formed. Okay. So when you see the cells, okay, the cells will divide into two. If you see a cell, the cell will divide into two. One has terminal cell and another one basal cell. I'll show you the picture. Terminal cell and basal cell. Then finally this terminal, no, it becomes a globular structure. It becomes globular structure. Then again it comes as a heart-shaped structure. Heart-shaped structure. Then two cotyledons are formed. Then only the seeds are formed. I'll show you the picture how it is formed. Now you can see the first one is the zygote. A picture is zygote. So the zygote undergoes a transverse division. So here you can see a transverse division occurs. Therefore a terminal cell and basal cell is formed. Terminal cell and basal cell. So upper is the terminal one and lower is the basal one. Then again it starts dividing. So now only the embryo is formed. So see the again the embryo divides. So four-celled pro-embryo is formed. Then the next thing is globular embryo. So the top you can see it is like globular embryo. The next one, the embryonal mass has become like heart shaped. So you have a globular shaped stage, then you have a heart shaped stage. So everything occurs in this development of this embryo, okay, dicot embryo. Then after that the mature embryo will have a radicle, two cotyledons. You can see two cotyledons. Radicle is present, radicle, then root cap, suspensor, everything starts coming. Finally, in a mature embryo, you can see the plumule also. So hope you know what is plumule. So radicle is the one which becomes the root system. Plumule, it becomes the shoot system. So these are the stages when it comes, when embryo is developed. Okay, again just to see all the diagrams. The zygote undergoes a transverse division to form upper terminal and lower basal cell. Okay, then further division occurs. So it reaches a globular stage, then a heart-shaped embryo. Then the mature embryo will have a cotyledons, two cotyledons, radicle and plumule. Okay, my dear students. So these are the stages involved in this. Now this seed, the fertilized ovule, is called as seed, and it possesses an embryo, endosperm and protective coat. As we already discussed, seeds may be endospermous like wheat, maize, barley and sunflower are non-endospermous like bean, mango, orchid and cucurbits. Now we have discussed how the endosperm is formed, what are the types and how an embryo is developed. Now we should learn about dicotyl seed and monocotyl seed, structure of dicotyl seed and monocotyl seed, which you would have learned in our earlier class in 10th standard. So it will be easy for you to remember. Even then we shall discuss once. The next topic will be polyembryony and parthenocarpy, which we can discuss in our next video. Learn well, study well. So do the breath exercise so that you can overcome COVID-19. Thank you all.